Hello, my name is Alaina Whelan, and this is my presentation of my biology lab final project. This final project is on the SARS-CoV-2 testing, also known as the coronavirus or COVID-19. My introduction. Where did SARS-CoV-2 originate? SARS-CoV-2 originated in Wuhan, China, and scientists were able to determine that SARS-CoV-2 originated naturally by observing the backbone of this virus, as a genetically engineered virus would have been constructed using backbone of a previously known virus. After seeing the SARS-CoV-2 backbone was substantially different from any known viruses, they were able to rule out genetic modification. Introduction. A virus is a microscopic parasite, much smaller than bacteria, that relies on a host to live and reproduce. Viruses rely on a host to replicate itself. Viruses also have the ability to mutate, making it difficult to find treatment. DNA viruses versus RNA viruses. A DNA virus has genetic information stored as DNA. These viruses are double-stranded and replicate inside the nucleus of a host cell. These viruses are first transcribed into RNA, and then mRNA is translated into proteins. These viruses also have a lower mutation rate and have large genomes. Some examples include HPV and hepatitis B. An RNA virus has genetic information stored in RNA. These viruses are single-stranded and are first transcribed, then replicated in the cytoplasm. These viruses are highly unstable due to its high mutation rate, making treatment difficult. They also have small genomes, and some examples include AIDS, SARS, and the common cold. SARS-CoV-2, or coronavirus, is an RNA virus, and SARS-CoV-2 is part of the Coronaviridae family, which is coronavirus. How does SARS-CoV-2 spread? This virus spreads from others infected with the virus. It spreads through small droplets from the nose and mouth that escape when a sneeze, cough, or someone speaks. These droplets can land on surfaces and can also spread through others touching these surfaces, such as tables, electronics, doorknobs, and buttons. It is important to maintain a safe distance, 6 feet, from others in case they are infected. It is also important to wash your hands frequently with warm soap and water, and you also should wear a face mask if you can. Currently, the outbreak status in the United States, specifically the southeastern Pennsylvania. According to the Johns Hopkins University of Medicine, there are currently 1,203,502 confirmed cases of SARS-CoV-2 in the United States and 71,031 deaths. The United States is currently the country with the highest confirmed cases in the world, and specifically in Delaware County, Pennsylvania, where my family and I reside, there are currently 4,269 confirmed cases of SARS-CoV-2 and 318 confirmed cases of confirmed deaths. Testing for SARS-CoV-2, PCR testing and RT-PCR testing. PCR testing stands for polymerase chain reaction, and it's a method in biology to quickly make millions of copies of specific DNA samples in a genome. This results in small fragments of DNA that can later be separated using electrophoresis, and I got this information from our lab manual. And then RT-PCR is a real-time reverse transcription polymerase chain reaction. It is a technique used to measure the RNA and translate it into a strand of DNA, to then use a PCR to multiply and get a measurable quantity to detect an RNA virus. Why is RT-PCR used for SARS-CoV-2? An RT-PCR is used for SARS-CoV-2 because an RT-PCR is used to detect RNA viruses, which SARS-CoV-2 is, and they are also used because they are faster and have been approved by the FDA in this specific pandemic. What would a positive result mean for a person with SARS-CoV-2? A positive result for SARS-CoV-2 does not rule out the possibility of bacterial or co-infections with other viruses in a patient. Patients' results are reported to the right public health authorities, and a positive result would mean the presence of SARS-CoV-2 RNA in the respiratory specimens and a detection of the nucleic acid from SARS-CoV-2. Testing for SARS-CoV-2, the ELISA testing. So the antigen-antibody relationship is the antigens are molecules that can stimulate an immune response. They also have specific features on their surface that allow them to create specific responses. Antibodies are Y-shaped proteins made by B cells in the immune system that respond when exposed to antigens. The specific antibody testing, there are 2 direct enzyme-linked immunoabsorbent assays, or ELISA, for the detection of COVID-19 IgG antibodies. And indirect ELISA, although there are multiple different techniques for this type of testing, an indirect ELISA technique is used to determine if you've been previously infected. This check tests to see if you have antibodies in your blood for SARS-CoV-2 and can detect if you had the virus in previous months. An indirect ELISA test has a primary and secondary antibody, and the well is coded with SARS-CoV-2 receptor binding domain, RBD, antibodies, and then washed. And then the primary antibody, IgG, is added and, if positive, attaches to the antigen. After another washing, the enzyme-linked antibodies bind to the antibodies. A final spike protein test is conducted, confirming the presence of the antibody. Final wash is completed, and then the substrate, called phenylene diamine dihydrochlorine, is used, and it turns yellow in the presence of the antibodies, indicating a positive test. What does a positive ELISA test mean for a person with SARS-CoV-2? A positive test means that the patient was both positive for the RBD screening and a confirmed positive by the final spike protein test, meaning they are positive for IgG antibodies, including an overall positive test result. Our problem to solve. Maura, David, Alexis, Johnny, Stevie, Roland, and Patrick decided to break quarantine and have a barbecue. Unfortunately, 1 week later, Johnny began developing symptoms of COVID-19, followed by Maura. All of the backyard barbecuers go to town and get tested using RT-PCR and ELISA. They're also blood drawn to do an antibody titer. Hypothesis of the test results. If Johnny began to develop symptoms of COVID-19 and then Maura after attending a barbecue with David, Alexis, Stevie, Roland, and Patrick, then the RT-PCR and ELISA tests for Johnny and Maura will be positive, as well as the others, because they were close to the infected people and they could have also been infected. These are the test results for the RT-PCR test. The test results from performing an RT-PCR SARS-CoV-2 test on symptom-showing individuals and their recent communications. Positive tests indicate a presence of RNA nucleic acid from the SARS-CoV-2 virus. It can be concluded that Maura, Johnny, and Roland are SARS-CoV-2 positive. The test results for the ELISA. Figure 2, ELISA test results from triplicate samples, concluding the purple wells as positive for the SARS-CoV-2 IgG antibodies, with both the RBD, receptor binding domain protein, and spike protein testing positive. It can be concluded that Maura, David, Patrick, and Roland are likely to be SARS-CoV-2 positive. And you know this because the purple wells mean it is positive. Test results for the antibody titer blood test. The results of a blood-drawn antibody titer test in relation to the individual shown symptoms of SARS-CoV-2, it can be concluded that Maura, David, Patrick, and Roland are likely to be SARS-CoV-2 positive due to their high number of antibodies. And then on the right is figure 3, and I used Excel to make this bar graph. And then this is how I made the bar graph. I then put this into my PowerPoint and it changed it to red to match. So my conclusion is that Johnny has a current SARS-CoV-2 virus because he does not have the antibodies for the virus, but he does have the RNA and nucleic acid from the SARS-CoV-2 in his body, meaning he currently has the virus. Moira, David, Patrick, and Roland have had a previous SARS-CoV-2 virus because according to the ELISA test, they have a SARS-CoV-2 antibodies in their systems. And then Alexis has not had the SARS-CoV-2 virus, and this is known because she lacks the RNA, nucleic acid, and antibodies for the virus. This is my works cited. And that's it. Thank you for watching.